S-Farm. Then similarly, the overall best PhD thesis, that's during the year 2007 and 2008 session in the University of Lagos. Ditto for the overall best for the Faculty of Science. And similarly, the Award of Excellence by Christian Progressive Cycle, CPC, of Yaba Baptist Church, Sabo Yaba. Then similarly, he was also recognized as the overall best student of the Department of Microbiology in the University of Lagos in 2008. And going furthermore on this, she got lots of grants, which include TET Fund, that's a Tertiary Education Trust Fund, and in some conference attendance, as well as some grants by the University of Lagos. Just to be brief on this. And to round this off, she's a member of the Nigerian Society of Microbiology, which is our major discipline, and the Institute of Public Analysts. And I believe we are all members as we are sitting down here, an Association of Industrial Microbiologists of Nigeria. Similarly, too, we have some of this association offshore, which includes the American Society of Microbiology. I've already mentioned S-Farm, and as well as the Organization for Women in Science for Developing World. This one now is also to even encourage most of our women to in areas of research and development. She has attended several local and international conferences, and her research work has been published both locally and internationally. So you are utmost welcome to give us a beautiful presentation. So while our audience will listen attentively for the very good moment of presentation. Thank you. So you will be on for 20 to 25 minutes, thereabouts.
Pleasure to be here this afternoon. I didn't know I would be the first person presenting today. I wasn't expecting it. I have two papers I'll be presenting this morning. I thought it was just a casual thing, but Ivan said, no, you cannot come here and just escape like that. So turning my paper presentation for the abstract session into proper paper. So the first paper I have is on bacterial and physicochemical assessment of potable water from all sub-presidents of the University of Lagos, University campus in Lagos Metropolis. I worked with nowadays research is a disciplinary attack. It's interdisciplinary, so we don't just work alone again. I work with people in chemistry department, Professor Oluseyi, Professor Dr. Fatusi, and student William Osuji. So we'll be taking a look at the physicochemical assessment of potable water in the University of Lagos. But before we go into that, what is water? Water is essence of life. Over 80% of the Earth's surface is made up of water. The human body has over 70% is made up of over 70% water, and even microorganisms over 80% of water. It's a natural resource. It's a natural resource, and one of the most important natural resources. However, the presence of the availability of water is a threat because the one that is used, the one that is available to us is the fresh water, is the one that is fresh. Water can be salty, can be from the ocean or surface water. The potable one that is available to us um, is usually found underground or in the surface and is threatened by a lot of factors, majorly due to human activities, our domestic activities, our industrial activities, agricultural activities usually threaten, contaminate the water environment. And that makes it very important for us to study, assess the water that we find in our various communities and see what we are taking if we are at risk from taking them. Um, the aim of this study was to investigate, just like topic, the physicochemical parameters of the potable water that is found within the hall of residences in University of Lagos. So we went around 15 halls of residence and we took the water at ransom, usually midstream. And we also look at the bacteria from this environment. We look at the bacterial load. We isolated the bacteria from this environment, and we try to look at their antibiotic profile of this bacteria. So, set sample collection collected from 15 halls of residence. The parameters, so chemical parameters that we look at were the total dissolved solid, dissolved oxygen, pH, salinity, electrical conductivity, and the temperature of the environment. And what we used in the assessment of these parameters was the Horriba U52 multi-parameter water quality meter. For the microbiological analysis, we did the total heterotrophic count, the total plate count of both the bacteria and the fungi. Even though we are more interested in the bacteria, we also look at the fungal load. And because it's potable water, we are looking at also residents of students take this water. We look at the coliform level. When you talk about potable water, you cannot but test the coliform level in it, detect it, and also enumerate it. Then we went further to identify the organisms that we got from these water samples, and the organisms were subjected to antibiotic susceptibility testing. Now the results. So, so most of the next slide. For the physicochemical parameter, most of the parameters tested fell within the acceptable WHO and EPA limit, even the some limits. However, we found that the pH of the medium of the water samples were way below. They were on the acidic end, which is far below what is expected by WHO standard. Also, the electrical conductivity was okay, dissolved oxygen. For some of the water samples, it was all right. Dissolved oxygen is an indication of a microbial load within the system. The electrical conductivity is dependent on the total dissolved solid, and they were both within this. All the other parameters, except for the pH and the turbidity, were outside of the limit accepted by World Health Organization. The next slide. That's for the parameter, physicochemical parameter. For the microbiological analysis from the halls of residence, found that the WHO limit is 1,000 colony forming units per mil of the sample, whether bacteria or fungi. And we found out that some of the halls of residence, the total bacteria count in there was above the acceptable limit, which indicates the contaminants within that environment. For the fungal heterotrophic count, some of the water samples had growth, whereas some had no growth, which shows that they were free of fungal contaminants. Next slide. Then we went on to do the coliform count, because we are talking about potable water now. We are talking about water. We do coliform count. It's an indication of fecal contamination. According to WHO standard, there shouldn't be coliform potable water. However, we recorded very high levels of coliform in all the water samples, and that makes them unsafe for drinking. So, next slide. So this is the MPN coliform results of the water samples from the all compared to WHO standard. So in some of the halls of residents, you can see the bars, the red bars, indicating very, very high coliform count, and that shows that these halls actually need emergency intervention to see to the water supply within the hall. The yellow bars less, and the green ones indicate safety. Then the bacteria that were obtained from the total count, we went on to isolate them, purify them, and identify them. And upon identification, we found we had 8 genera of bacteria. Next slide. So we had 8 genera. We had Klebsiella genera, the genus Klebsiella, Escherichia, Enterobacter, Pseudomonas, Proteus, Staphylococcus, Providentia. These were the genera we isolated. And in order of distribution or occurrence in the water samples from the hall, we found that we had more of Klebsiella, Escherichia, and Enterobacter. You know, earlier on we said that the coliform levels were high. Klebsiella, E. coli, that's E. coli, E. coli where we enter bus, E. coli, Staphylococcus that is spread out. So they were very high. So the very high values, they all came from coliform samples. So that really indicates that those water samples were really, really contaminated. Then we went on to do the antibiotic susceptibility test. So some of the organisms were gram-positive, like the Klebsiella. All the coliforms were gram-negatives, and the gram-positive one was the Staphylococcus aureus. So we tested them against some antibiotic disc in the laboratory. So let's see the results. So the results I have. So there was a general resistance to Ampliclox, a 100% resistance to Ampliclox. So the isolate, the granular Ampliclox, cefotaxime, cefuroxime, all of them, the organisms they showed a 100% resistance to them. For the bacteria, there was complete resistance to amoxicillin, Ampliclox, and ceftriaxone, for full sensitivity to ciprofloxacin. Meaning, if somebody is infected with for the gram-negative, please don't administer antibiotics without seeing a doctor. I believe we all know that. The only antibiotics that will work for you is the ofloxacin for gram-negative and ciprofloxacin. And I know that when we are sick, we just go to the pharmacy. I have this, I'm purging, give me antibiotics, and you just buy antibiotics off the counter and administer. And we will know that the antibiotics we actually apply does not have an effect if it's a 100% resistance to Ampliclox. I remember there was a time, anytime we go, Ampliclox is the solution to all illness. The Ampliclox no longer works. So what we are just doing is we are just building antibiotic resistance and increasing antibiotic resistance in the environment. Okay, so this table shows the susceptibility pattern of the gram-negative bacteria to the various antibiotics. So we can see the Ampliclox, the ACX, that's the 100% resistance, and we have them to 100%, 96, 64, 87, 58. Those are very high resistance. Meaning, if we go to the pharmacy and buy antibiotics over the counter, they will not work for this isolate. The infection will just persist. Okay, so the next one too is also showing susceptibility pattern to another set of antibiotics by the gram